Good afternoon, everyone. My name is Dr. Jeff Thomas. I'm one of the physicians here at Health Coach 7, and I just want to thank you all very quickly for being a part of this introduction tonight. One of the things I want to bring to your attention is that if you've been following us over the past few weeks, Dr. John Catanzaro has done a wonderful job of outlining all the various metabolic processes and the relationship to methylation. A few weeks ago, I was a part of one of those segments and went ahead and discussed briefly the relationship between detoxification and methylation. Last week, Dr. John went into a bit of a cursory review over that of the gut and methylation. And as he continues on his segments, I thought I would go ahead and follow up with that in a much more sort of in-depth way of looking at the relationship not only between methylation and gastrointestinal health, but the microbiome as well. What is that, and what is its relationship to these other aspects of our health? And so this afternoon, that's what we're going to go ahead and discuss. And so what I'll do very quickly is give you a brief outline as to each of the objectives I hope to cover this evening. And then at the end of this, provided we have time for questions, we'll go ahead and entertain them at that time. If you have any questions, please feel free to go ahead and email them via that chat option, I suppose, as well. And I will address them at the appropriate time towards the end of the conversation. But with your permission, I do ask that you allow me to get through pretty much the bulk of the information just to make sure that I'm able to cover everything. And then at the end, we'll go ahead and address those questions. So with that being said, here's what we're going to discuss as an overview this afternoon. First off, we're going to start with just some basic knowledge. What is the anatomy and the physiology of the gastrointestinal system, which from here on out, I'm just going to refer as the GI system. And then we'll go ahead and dive into the relationship between methylation and that of the GI system. Some of those common conditions that we may or may not have seen that affect methylation as well. From there, we're going to switch gears and talk about this microbiome thing. What is that? What's that about? What's its relationship to everything else we've discussed? And then we're going to shift back a bit to some of the signs and symptoms of that in relation to disturbed methylation within the gut. Once we've got a big picture of that, we're going to shift gears and look at those genes that are involved in GI health, or at least in the assessment of it. In that, when I have a patient in front of me, for example, and I have their Health Coach 7 genetic report, what am I focusing on and why? And then we're going to look at some of these gene SNP variants and how they actually can potentially affect GI function. From there, we'll discuss some of the examples once again of when I have the report in front of me, how I interpret that, and then we'll follow that up with some treatment approaches. And then at the end, we'll go ahead and look at some questions if we have time. And so with that being said, let's go ahead and dive on in. And the first slide that we're going to look at right here is a very detailed breakdown of metabolism within our gut. Now, my undergrad was in nutrition, and we actually had to draw this as one of our, how shall I put this, one of our finals for a class that I had to take. And I'll be honest with you, that was a very time-consuming project. But in the midst of all of that time, I really got a sense of just how miraculous our body is in terms of how it's able to break down, assimilate, and utilize various nutrients. What's really cool is when you look at the differences between carbohydrates, fats, and proteins, how does our body recognize them? How does it break it down? How does it use them? And as a result, what is the energy that is derived from each one of them respectively? And so very much so in an over-like fashion, what I just want to simply point out with this slide is that the metabolism that takes place within the gut is a very detailed process, is a very complex process. And so as a result, that kind of investigative process that I have with my patients can take a bit of time, because what we really want to assess is basically what is the obstacle to cure here. And by understanding how metabolism works within the gut, you can see that our search can go many different places. And so that's just briefly what I want to go ahead and share with you as it relates to this slide. And so jumping right ahead, what does this all have to do with methylation? Well, what you can see before you is essentially a diagram that's illustrating homocysteine metabolism as it relates to methylation. You may or may not know, but homocysteine is a very sort of classic piece within the methylation system. As you can see, when we start with proteins, we're looking at here in that circle within the center is essentially the activation of methionine, and so how that's recycled due to various cofactors from homocysteine to methionine, and as a result, how that go ahead and produces molecules within the folate system as well as sulfite products as well. And so just so you can understand from this particular diagram, the relationship between the GI system and methylation is that most of our nutrients and molecules are going to be derived from essentially some aspect of nutrition in order to facilitate this process. Now, what are some of the conditions that we are going to look at that may be affecting methylation? Now, as you can see, here is a very comprehensive list of just some introductory items, some of the more, shall we say, popular aspects of gut conditions that tend to affect methylation within that system. And so whether it be small intestinal bacterial overgrowth, as well as various forms of infection, we definitely see a fair amount of yeast. Also, for some of our more, how shall I put this, busier clients, those who need a pleasant reminder to eat from time to time and not do it in their car on their way to work, or not only do it once a day. As a result of those people who tend to that particular lifestyle, it may be an inadequate nutrient intake that could be contributing to their inability to methylate, as well as just due to the fact that the world we live in, despite our best efforts, is going to be that of a polluted one. And so depending on your sensitivity, where you live, were you raised on a farm, do you live next to a plant of some sort, do you live next within the city even, for example, and getting exposure to things such as electromagnetic frequencies, things of this nature, really looking at the whole gamut of potential exposures and looking to see if for you especially, is this going to be an issue? Once again, stress can certainly affect methylation, so looking at the sources of stress within someone's life, but not so much that they try to avoid it, but they're able to, in a healthy-like fashion, manage it. Going down to some autoimmune diseases, certainly celiac is going to be a condition that can affect methylation, and then just some more lifestyle aspects, sleep, exercise, and then just some sort of, how shall I put this, the presence of some sort of spiritual enrichment, something that really allows you to tap into something greater than yourself and can be a renewing experience. That's just something that I definitely check in with my patients for, because I have found that for those that do have this practice within their life, however that may look, they tend to have a much more healthier outlook and as a result a healthier lifestyle. So here are some other examples of those conditions and their relationship to the gut. Now we'll go into this a bit more in detail as we continue our conversation, but certainly autism, I believe, is a really good example of how we have this connection between our gut and our brain, and how when one of those is, how shall we say, out of balance, can certainly affect the other. So on the left-hand column here, you can see that within a quote-unquote healthy or normal individual, what their gastrointestinal function is going to be looking like, as well as some of the functionality of the brain, as well as that of the activity of the immune system. When we look over to the autistic spectrum and those that present in this fashion, we're going to see that there's going to be compromises within the gut, as well as an alteration within the production and the metabolism of neurotransmitters, as well as some of the issues within the gut, that especially of the microbiome, which we will go into here in a bit in the next slide. And so, excuse me, the slide after this one. Here are some other diseases of the gastrointestinal tract. And what's important to understand is that in some way, shape, or form, should one of these have been present or is present currently, we certainly want to see, is this in some way, shape, or form affecting that person's ability to methylate? And if so, we'll go ahead and address it. But either way, you can see that there are many different conditions that can ultimately affect the GI system. And so it's my job as the facilitator of your health to make sure we're ruling out any of these serious conditions. And once we identify what the underlying cause of your particular signs and symptoms are, to address that appropriately. And so jumping over to the subject of what is the gut biome, basically we have this really fancy picture here. And what this is pointing out is that in addition to the 21,000 genes that we have in our body, we have this family of bacteria, fungi, viruses, and archaea. Now archaea, basically their involvement is going to be in the digestion and the fermentation and breakdown of plant fibers. So for those of us who eat any kind of vegetables within our diet, they're certainly going to be an important part of that digestion process. With that being said, this collective species of bacteria, fungi, viruses, and archaea are estimated to have about 100 trillion different organisms within our body, in addition to these 21,000 genes. Now, what's fascinating is that this microbiome in and of itself is said to contain 4.4 million genes. So you have to think about that for a moment. We have that is associated with our cells, those genes, and then we have the genes that are a result of this microbiome. And when you look at the population of genes as it relates to the microbiome, as compared to that within our human cell population, you can see that for every 10 genes that we have one within the human cell population. And so our microbiota outnumber our human cells by about 10 to 1. When you think about that, another way to put that into perspective is that the total population of bacteria could total anywhere from about 2 to 5 pounds in overall weight. Something about that just absolutely blows my mind. And so when we're wanting to address the microbiome, this is certainly going to be a very considerable aspect of our overall existence. But why? What is this about? What does it do? How does it get out of balance? And if it is out of balance, how would we even know? These are all really great questions. And so what we want to go ahead and discuss right here is the purpose of our microbiota. And so as you can see here in this list before you, there's many different aspects of the microbiota that are going to be critical to our day-to-day existence. Everything from digestion to the assimilation of energy and nutrients, to boosting our immune system, also promoting and preventing, for that matter, things like leaky gut. For those of you that may not have known, they can also influence mood and behavior. But more specifically, which I find rather fascinating, is that they're responsible for the production of anti-inflammatory factors. They help with pain relief. They help with pro-oxidant defense systems as well as nutrition. Now, when this particular balance within the microbiota of, say, good and bad bacteria becomes out of balance, what can happen? Well, there can be a deregulation of genes that are involved in energy metabolism. And as a result, there could be an increase in the production of toxins, and those toxins can ultimately affect DNA to the point that where there are mutations observed. And those mutations can certainly have an impact on nervous and immune function. And so clinically, what does that mean? For those people who are coming in here that have a long list of chronic disease, who are dealing with obesity, diabetes, or even cancer, what we're going to want to look at is their microbiota. We're going to want to see what is the population of the good guys in relation to the bad guys, and how can we balance that? What is contributing to that imbalance in the respective populations within that? So how is one to know when they are experiencing some aspects of either disturbed or imbalanced microbiota, as well as that of disturbed methylation within the gut? Well, let's go ahead and talk about first those aspects of a disturbed system as it relates to methylation within the gut. So any of these particular signs and symptoms, and there are many more that are not included on this list can certainly contribute to altered methylation within the gut. And so should any of you have experienced these at one point in your life or experiencing them now currently, we are certainly going to consider within our differential diagnosis the possibility that there may be some methylation impairment. Now, to some degree, I do believe it's safe to say that we all experience this, although for some of us it's much more apparent than others. And so not to say that one person is going to be far worse than the other person. I believe fundamentally we are all experiencing methylation disturbances to one degree or another. Unfortunately, some of us just have to deal with it a bit more so than others do. And so looking at the genes now, when we are wanting to focus more so on the genes involved in assisting of GI health, this is just going to be a list of various genes that I consider in my assessment as it relates to GI health. So starting with the first one, COMT, this particular gene can either be downregulated or upregulated. In the effect or in the context of it being downregulated, what we tend to observe is that there could be a backup of neurotransmitters, more specifically that of dopamine, epinephrine, which I may refer to as adrenaline, and norepinephrine. Should we find that COMT is upregulated, then there's going to be an increase in the presence of neurotransmitters. And so what we want to make sure is that in order for COMT to function appropriately, there needs to be the presence of methyl donors. And so that's why we will go ahead and supplement with various methyl donors should we find that there may be a mutation within COMT. Now, what are some of the symptoms associated with someone who may have a COMT mutation? Agitation is a big one, and that's usually due to either an increase in the production of adrenaline or in the degradation and the increased degradation of adrenaline. And so we want to make sure that if that's present to any degree, why that is, and address that respectively. And so the goal overall is to have COMT be in equilibrium. We don't want it too far upregulated, but we also don't want it incredibly downregulated either. So it certainly is a balance. Moving on to MAO, monoamine oxidase. This particular gene is involved in the detoxification of neurotransmitters. When we find that this gene is upregulated, it can lead to increased degradation of various neurotransmitters, thereby leading to clinical aspects of, say, depression, for example. So if someone presents with depression, I'm certainly going to want to look at MAO. Moving down to MTR, this is methionine synthase. This is involved in the recycling of homocysteine and the production of SAMe. And so once again, if we find that an individual is having elevated high homocysteine, we're going to want to take a look at, say, for example, is their MTR obviously mutated? And if it is, how can we support a healthy, more balanced homocysteine metabolism? And so we have SULT, which is a phenol sulfotransferase, These genes are responsible for supporting phase 2 detoxification, specifically sulfur metabolism. So for those people who are having a sulfur sensitivity, we're going to want to take a peek at this, and if we do find that there's a mutation in that, we'll be able to address that specifically as well. We do find that for more often than not, a lot of people who are having heavy methyl disturbances, as well as presenting with a long list of chronic diseases, that the SULT is certainly going to be mutated to some degree. Now, the MGAM, the maltase glucoamylase, this is one that pops up for a lot of people who have the possibility of being starch sensitive. And so when I see that one pop up as red, which would be homozygous positive, we certainly want to evaluate the possibility that there may be a sensitivity to starch and or wheat, and if so, want to go ahead and evaluate that closer. The HLA-DQB1 is involved in the immune system, but we do find that there's a strong connection in those that have a mutation in this particular gene and the possibility of celiac disease. Once again, an individual here who may have a sensitivity to wheat, and so we'll take a closer look at that as well. Right here beneath that, we have leptin and leptin receptors. This is going to be involved in the hunger response. Those people who are having, say, an imbalance or a challenge in their appetite control, appetite regulation. And so when someone presents with someone who, say, has a habit of maybe, say, overeating and is not able to have more frequent, not frequent, that's not what I'm trying to say, but a more disciplined routine time of eating throughout the day, but just tends to overeat throughout the entire course of the day, we may want to take a peek at that as well. And the last one is the CTLA4. We'll also see that one pop up, pardon me, for individuals who may be sensitive to things like wheat and or gluten. And so these particular genes are what we take into consideration when I'm wanting to assess an individual's GI health. Now, when these particular genes are presenting themselves, what are some of the things that we want to be concerned about clinically? And so we've already discussed this to some degree, but just to put it all together, essentially there's going to be some impairment typically in neurotransmitter metabolism. For certain individuals, this may be contributing to somewhere or why they tend to be falling on the autistic spectrum disorder. Their children may be observing ADHD. For certain individuals, they may have various presentations of behavioral and or cognitive issues. We've already talked about depression and anxiety. Another thing we haven't really gone into detail about is that those people who exhibit compromises in histamine metabolism. So these are going to be people who are going to be much more hyperallergenic. And so they're going to be much more sensitive to allergies and insults that may just provoke some degree of an immune response that makes them overall very uncomfortable. Overall, though, what we can expect is that when we are observing various mutations within gene SNP variants, that there are going to be compromises in gene expression. So what does that mean exactly when we break it down like that? Is that as a result of there being compromises within the gene expression, basically there's going to be certain genes that can be turned on or off, leading to a variety of ways in which that particular gene is expressed. And more often than not, what we suspect sometimes is that that just may simply mean there's an increased susceptibility to various chronic ailments. And so let's go on to the next slide here. I bring up the vagus nerve because when we want to look at the relationship between the gut and the brain, this is something that I'm not too sure a lot of people are aware of. But as we've been talking about now, the production of neurotransmitters and the balance that we're wanting to maintain between adrenaline, norepinephrine, serotonin, and even some aspects of sex hormones like estrogen, when we're looking at the gut, there is a production of various neurotransmitters that occur within the gut, and they are released from the microbiome. Those bacteria, those fungi, and those archaea that we discussed can release their own neurotransmitters, just like our neurons do. And those neurotransmitters can communicate with the brain in a similar fashion that our neurotransmitters in the brain do, and they do that by way of the vagus nerve. And so I bring this up because clinically, when I see someone, for example, who is living a very, say, high stress life, and they tend to really embody or really hold on to that stress to the point to where they're presenting with a whole host of various gastrointestinal symptoms, whether that be abdominal pain, cramping, bloating. They may even get really nauseous. There may be some vomiting to some degree. Bowel movements may be a concern, whether that be in the context of diarrhea or constipation. What I really try to step back and look at first and foremost is what are the potential aspects within their life that are contributing to impaired GI function, that are creating things like anxiety and or depression. And typically it's some sort of external factor that's present in that individual's life. And so while they may be focused on the anxiety and the depression, say within their brain, what I want people to understand is that sometimes this can actually be stemming from a gut issue. And so when you come in here and you're concerned about mood disorders, we're going to talk about the gut because there's a very strong connection between those two. And so that's just something that I wanted to share with a lot of people because I'm not sure how common knowledge that is. And so this Health Coach 7 assessment right here, this right here is an individual who's coming in just wanting to know their overall genetic risk report, if you will. And so as you can see here, we're dealing with a lot of yellow. We're dealing with someone who's heterozygous, positive and negative. Not too much on the red spectrum, not too much on the green. So they're kind of right there in the middle of the road. And so if I didn't know anything about this person and I was only looking at their genetic report, based off of what I'm looking at right here, from what we've discussed earlier, I would certainly say that this is an individual who I believe is probably to some extent going to be dealing with some sort of mood imbalance. They may find themselves either prone to stress or prone to depression. Now, what I will mention to you, just because I actually know who this person is that this was an individual who has a history of bipolar disorder. And so it's interesting to see that we're seeing the presentation of COMT and MAO pop up here because this is an individual who tends to fluctuate and vacillate between both ends of the spectrum, between anxiety and depression. And so it's pretty interesting to see how that presents on their genetic report. I also know that this is an individual who has a history of being sensitive to starch. And so what we can see here right at the top is HLA-DQA2 as well as MGAM down here. And so just knowing that here is an individual who is dealing with some pretty strong characteristics of mood disorders and has a known sensitivity to starch, clinically what I would want to do is remove those insults, those threats, those particular aspects of their life that may be contributing to this hormonal imbalance, this neurotransmitter imbalance, and see if we can begin to modulate the responses just with that first and foremost. Now also we're probably going to want to really go ahead and support the sympathetic, or pardon me, the parasympathetic system within this individual, just especially if they tend to be more manic. I bring that up because once again, this is a patient that I worked with who I know has a history of mania. And so knowing that, we're going to want to look at various ways that we can support more of a parasympathetic or a more relaxed, calming state, if you will. Now here's the remainder of their genes as it relates to the GI assessment. And so you can see that we have the leptin receptor genes popping up as well as CTLA4. So I'd want to take a peek at this person's eating hygiene as well as their body composition to see if maybe the leptin diet may be something that we would want to recommend for them. And as we've already mentioned, they've had numerous presentations with the MGAM and the HLA-DQA1-2 at the top here, as well as what we're seeing, the CTLA4, with some sensitivities to starch and as gluten as well. And so we'd probably want to explore that further just to see if there would be any relief from that provided with that being the case. And so what's important to remember is that as we've discussed all of these aspects of methylation and the microbiome, mutations in methylation do not lead to certain medical conditions. We're only talking about the increase in the susceptibility. So it's important that when we look at this person, how do we really put this all into context? Yes, we just saw a person's genetic report that on paper, you could say had some concerning aspects to it, but I had also mentioned to you that I was aware of their clinical history as well. So once again, wanting to look at the whole person and not just making them out to be just a genetic report. So how do we do that when we approach things from a treatment point of view? So what I want to do is take into consideration that yes, here is an individual. We'll stick with the example that I gave you on the Health Coach 7 assessment. Here is an individual that presents with bipolar disorder who has a strong possibility of a sensitivity to gluten and wheat. And so nutritionally, what we're going to want to examine is just before we make any adjustments, what does their diet look like? What time of day are they eating? What are they eating? What's their relationship to food? Do they eat when they're emotionally unstable, or do they eat to calm themselves? Do they eat when they're stressed? Do they forget to eat when they're stressed? When they are stressed, what do they eat? When they want to be comforted, what do they eat? As you can see, this is a very long list of questions, and the idea is to really understand this person before I make any judgments and or recommendations, but to really get to know why they do the things that they do. Not just what they do, but why. And then as a product of that, I can help them to understand the pros and cons of their decision making and help them to educate them as to whether or not in the future they still want to continue to make those decisions. Once again, as it relates to physical activity, I'm certainly going to educate them on how that can be beneficial, as well as the various ways to manage stress, and talk about the importance of hydration and sleep as well. So these are what we call the pillars of health. These are the foundational aspects of health that I consider to be critical before we make any other recommendations. This is what we want to optimize first and foremost. Once we've been able to optimize that, if we haven't been able to achieve the results that we want, then we'll take it a step further. But if we have, then great, because what we've done is take a lifestyle-based approach to addressing this person's condition, and hopefully if we've been able to improve that, then fantastic. I do think it's important to remember that these things, these aspects can be beneficial and can make a profound difference on your health. Moving on to addressing of the gut health, we have been talking about the GI system. So certainly if we're finding that where there's an impairment in methylation within the gut, we're going to want to address those impairments, whether that be due to a nutrient deficiency or some other aspect of, say, autoimmunity. We're going to want to try to address that and support that. That way we can restore vitality. We're also going to want to look at the microbiome. What does the balance within the microbiome look like? Are they finding that there's more of a population of the quote-unquote bad bacteria and not so much of the good? And if so, what are the ways that I can support this person to have a more balanced microbiome? And then with some of the nutritional recommendations in the way of supplementation, whether that be with IV therapy or oral supplementation, whatever the case may be, we're going to do an outside-in approach. We're going to do a well-balanced approach to make sure that we're addressing methylation as well as all those cofactors that are necessary and supportive for methylation as well as metabolism. So with that being said, I want to thank you all for listening. I hope I've been able to really share something that you've learned that was new and worthwhile to your own efforts to sustain health and wellness. I look forward to working with any of you in the future. And so if you have any questions or would like to schedule your own GI assessment, then I would like to guide you to this particular slide right here. We're open for appointments right now as we speak. So if you're interested, please give me a ring, as well as if you have any feedback. This is something I would like to continue in the future, but I certainly want it to be on subjects that are going to be relevant and important to you, and done so in a way that's actually going to facilitate learning for you as well. So with that being said, I look forward to hearing from you, and if you have any questions, now would be the time to go ahead and shoot them to me. Once again, thank you so much.